We are eukaryotes, descended from the first eukaryotes, knit of all the features that we share.
Human cells contain a great diversity of kinase enzymes belonging to the kinase gene family. Many of these can be organized into subfamilies, such as the MAP kinase subfamily, the casein kinase subfamily, and many others.
In yeast, there have also been a large number of duplications of kinase genes in the gene family, producing a number of subfamilies such as the MAP kinase subfamily, the casein kinase subfamily, and others. There are a few which belong to yeast-specific gene subfamilies, but the majority of these kinase enzymes belong to subfamilies found in higher eukaryotes. The first eukaryotes seem to have diversified their kinases in ways which are shared among the eukaryotes today, such as larger subdivisions of the MAP kinase pathways with more regulatory power, the use of casein kinases to respond to ultraviolet light for circadian rhythms and to be involved in the cell cycle, and many others.
The protist group of choanoflagellates are thought to be the closest protistan relative of the animals, and we are the only protists to possess receptor tyrosine kinases, a very important gene family in the animals, but nevertheless it seems to have evolved in eukaryotes just prior to the rise of the animals.
A great diversity of proteins regulating the cell cycle are shared in all eukaryotes, and homologs of yeast and human proteins exist. This includes a number of the checkpoint proteins which can stop the cell cycle, and perhaps the homolog of BRCA. Although yeast and other protists can be unicellular, nevertheless, there is programmed cell death which can be triggered in response to certain stimuli, such as excess pheromones. And a number of the proteins which animal cells use to induce apoptosis can also then kill yeast cells by triggering apoptosis in yeast, such as BAX, some of the caspases, P53, APAF1, and others.